mainly job opportunities, research opportunities, or similar things. So one of the things I would like to tell at this point is that, probably 40 years back, things were very different, especially biotechnology, biopharmaceutics, biochemistry, microbiology, pharmacology, pharmaceutics. So 80% of the opportunities were in teaching or research. And that was the time most of the people used to go to US. After teaching in MS University as an assistant professor, as soon as I got my PhD, I went to US. After my postdoctoral, then I was in UK for the next 13 years. I was teaching. Then I came back to India. That was the time the opportunities came in life sciences, biopharmaceuticals. Pharmaceuticals came up, and I have joined as the vice president and head of drug discovery at Sun Pharmaceuticals. Now, there is a big shift was there when I was doing my PhD or one my student time, and now the opportunities are enormous. Whichever, or even the art students has a tremendous opportunity. And what I wanted to tell here is that whatever I am going to talk mainly the research opportunity, which comes only 20-30%. Please remember, right now, as I mentioned, 60-70% is other opportunities. For example, a tremendous opportunity is there in IT industry. Many students in biotechnology, biochemistry going to IT. Many are going into management, production, marketing. So this comes about 50 to 60%, and quite a lot of people after after biotechnology they go for an MBA to be management. They're managing whether it is research institution, whether pharmaceutical industry. There is some voice is coming from the background. I think someone is talking. You have to put the mute. So things have changed now. People have got enormous opportunity, and a major opportunity came is entrepreneurship. I don't think people ever knew what exactly is entrepreneurship, especially how to get money, how to set up a company, how to initiate, how to value a company. All those things were, at least I never knew about it when I was studying or even later period. All these things have come in the last 10 years. So let me give an insight into the opportunities now based on life sciences or biopharmaceuticals, pharma industry, etc. You can see the right hand side, multiple companies. These are all startup company, very successful startup company. Few are in Kerala, Bangalore, and in Chennai. And so we also have overseas facility also. One of the companies, Sai Genome, which is based at Cochin, in Aziz, that's on. So basically involved in, it's an incubator facility, others can incubate, and few other research is going on. Saxon is one of the earliest company which I started, which is mainly involved in the development of biopharmaceuticals, and we have developed a molecule, put it into phase 1 trial in the US and in India, mainly a Fab molecule for macular degeneration or age-related macular degeneration. And first in kind, which is developed from in India, took about 3 to 4 years, and probably we spent several $millions, but the valuation has gone zoomed multiple times. And these type of opportunities are enormous, but you have to look for. Theragen Life Sciences is another similar company which is mainly involved in the development of molecules for diabetic retinopathy, a similar molecule, a molecule which is inhibiting the angiogenesis process. Another company is a nanotechnology company called MagGenome. MagGenome is mainly involved in the development of magnetic nanoparticle based products, which is for DNA extraction, RNA extraction, protein extraction, antibody extraction, cell extraction. And recently, all our sales went through zero when the COVID started. This MagGenome, by the way, the MagGenome, the entire technology is absolutely a novel technology, patented technology. Then during this pandemic, in the last 4 or 5 months, we have developed viral transfer medium, molecular transfer medium, RNA extraction kit, and RT-PCR. Please remember, all these things what we have developed is a totally novel methodology, and we got the approval. And this month we have over 1 crore sales has happened for single product. So that's an opportunity we have utilized. We never thought of developing an RNA, a novel technology-based RNA extraction using our technology, but then an opportunity came, we used it. Imagine. So everyone has got such opportunities. You need to know how it can be used. Another company is called Theragen Life Sciences. Theragen Life Sciences is mainly involved in the generic product development for international market, mainly US and Europe, called ANDA, abbreviated new drug application. I'm sure that MedGenome, everyone knows, MedGenome is one of the most successful company in our group. In 4 years, it became more than 400 million, about 800 people are working, and mainly involved in next generation sequencing. Next generation sequencing has never come about 4 years back in India. It was started by our chairman. So it's, I think all over India, and we have extending that, that MedGenome to other countries, especially in the US. Theragen Molecular Innovation is a new company which we have started now. Mainly IgY, immunoglobulin Y, or chicken-derived antibodies, which is from egg yolk, and various opportunities are there. Now, this is a big project we brought from US, and we are trying to develop a molecule or an antibody, an orally active antibody, first in class in the world, and probably such a molecule first time might be going to US FDA for alcohol-induced hepatitis, and a similar molecule we are going to develop for Clostridium difficile associated diarrhea, as well as for inflammatory bowel disease. So these are some of the successful company in the few years, and out of this Theragen Molecular Innovation, which is started very recently only, we also have got Cygenome Life Sciences Private Limited, which we were supposed to start in the facility in Kerala, but it is pending. We also have biotech biopharma skill enhancement program. By the way, this is the 20 students every year we take, but last 1 year it is pending, and this is funded by Kerala State Industrial Development Corporation. Our idea is to train students after the post graduation, develop their skill set so that they are employable. So these are some of the things what we are doing. Now here, what I want to tell is that, what are the opportunities from thousands and thousands of years back, and how that has improved over a period of time, and what you can really do, or what are the types of industry what you can look into. I'm not going to tell you can apply to Sun Pharma, or you can apply to Biocon, or you can apply to Syngene or our company. That's not what I'm going to tell. I'm just going to tell these are the opportunities available worldwide in the various area, and that also I am precisely and concisely telling about in the research area, not in production or manufacturing or quality control or management or marketing. That's not the thing. So that, and IT. So mainly research, what all things can be done. Now, what exactly is biotechnology? Everyone knows that in single word, how living organisms can be used, or the products of living organisms, such as an enzyme, how this can be used for human benefit or animal benefit or its application. That is basically biotechnology. That is probably one of the definition or explanation that can be given. And what exactly is classical biotechnology? Classical biotechnology has not started now or 100 years back. Probably few thousand years back it has started. Now, all the production, what is happening now, products has some connection with the classical biotechnology, which you will be able to understand. But whereas what exactly is modern biotechnology in the last 30, 40 years, probably, and especially that's 20 years, significantly which has come, mainly the use of the bacteria or the cells or the DNA or the RNA or the functional protein, how this can be utilized, how you can manipulate, and various products can be developed. This is what is the modern biotechnology. Now just see some of the classical biotechnology. Everyone knows that, everyone knows how to make idli, dosa, khaman, those type of products, fermented products, isn't it? Yogurt, by the way, you just have to understand one of the greatest fermented product, the successful product is Yakult. Yakult is a simple probiotic based fermented tasty small drink which is currently selling about 15 billion. Imagine somebody got an idea to develop a probiotic, but you see the background, the probiotic, that particular bacteria is patented. 15 billion. That means the valuation comes to about 150 to 200 billion dollar. And what they are selling only single product worldwide. How it has come, your more early, I mean, the classical understanding that is, and really translated into a modern product. So the production of food, like curd, probably Indians used to maybe 5,000, 6,000 years back, or even 10,000 years back, curds were used. Then Iraqi started using cheese. Cheese needs fermentation process as well as certain animal enzyme. Certain enzymes like lipases, even some proteases, etc., used for the cheese production. Wine, right? Everybody knows about alcohol, how it is produced, various types of alcohol, and especially, distilled alcohol. But then origin is from wine. Wine from which was started probably about 5,000 years back, and probably Romans started developing this one. Then Egyptian started making bread, probably 4,000 years back, and even now we are using. Probably there is not great improvement in the technology, except the technology, the yeast is probably same, improved yeast versions are there, but also preservation method has significantly changed. That is all what has happened in the last 100 years. And classical biotechnology was also used for the treating purpose. An example is that molds were used for curing or inhibiting bacterial growth. And the latest, if you remember, about 50 or 60 or 70 years back, the penicillin, which is also a product of microorganism. Now, these are some of the advances in fermentation technology. Today, many things are produced. Now, I mentioned about some of the food and food products, what we are using, but that's not the end of it. If you really go and see, everywhere there is an opportunity, a background and application for biotechnology is there. Pharmaceutical compounds such as antibiotics. Now, everyone knows about antibiotics. Some of the earliest antibiotics as totally by fermentation process. Subsequently, part of the antibiotics by fermentation and synthetic procedure or semi-synthetic. Now, please remember, not only antibiotics, but many of the NCs, new chemical entities, molecules are also produced via fermentation process. And also many of the immobilized enzyme, or what is called the green chemistry, is used converting one pharmacophore into another one. You don't need to use solvents and other things. You can use enzymes. The enzymes at normal temperature can convert one product into another. That's another upcoming field in the pharmaceutical industry. Everyone knows about single cell protein. Proteins is by fermentation process, and amino acids. Amino acids is widely used. All the amino acids mostly produced using biotechnology techniques, very rarely by synthetic method. The same way, various chemicals, hormones, and pigments are used by fermentation process. Enzymes with a large variety of uses. There is enormous applications are there. Enzyme cannot be synthesized. It is a fermentation process from the bacteria, mammalian cells, fungus, etc. is produced from enzymes, and enzyme has multiple applications. As I mentioned, biotechnological application where bread production or cheese production, all these things, and in agri-foods, enzymes are used. So, I mean, enormous opportunity. There are hundreds and hundreds of companies which produces enzyme. Then biomass for commercial and animal consumption, such as single cell protein, which everyone is familiar with. Fermentation and genetic engineering have used for food production since 1980. So all this has happened last 20 or 30 years only. Before fermentation came, for example, phenylalanine, many of the amino acids were used by extracting from plant or animal foods, by hydrolysis and separation, by chromatographic techniques. So this was the technology which is used. Now it is changed. Now genetically engineered organisms are cultured in fermenters to produce large quantities of desirable enzymes which are extracted and purified. Now let's see that classical biotechnology which is used in insulin. Everyone knows about diabetes, whether it is type 1 or type 2. And the drugs for insulin, drug for diabetes or resistance to the insulin, it has come only 40 or 50 years back. Virtually about 100 years back, there was no drugs for diabetes at all, whether it is type 1 or type 2. Eventually, people get various kind of nephropathy or, the diseases of the eye, heart. Many problems used to occur. Early death was there. But in 100 years, things have changed. So that is what sequentially I have given. What I wanted to tell here is that in the last 100 years, from nowhere, we started extracting insulin from animal source. Then we have significantly changed that process that I will be able to show that. Just I wanted to also tell, insulin was originally produced in University of Toronto. In fact, I have seen that lab over there. They have kept over here. So insulin was University of Toronto only it was kept. So, but things have changed in the last 20, 30 years because of the advent of molecular biology, biotechnology, and its use, especially recombinant DNA technology. Here I am again giving as a simple example, one of the most used biotechnology product is insulin, where we were in 1992 and how things have changed. And probably by 1990 to 2000, modern insulin start coming. So what was the difference earlier? Earlier it was extracted. Earlier it was semi-purified. You have a lot of immunological problem, and probably back in 1973, only single chain or single unit of insulin was extracted without other impurities. And earlier there was a lot of immunological problems were there. Then 1980, recombinant human insulin. Recombinant human insulin is the product of one of the earliest, or what you can say one of the first product of the modern biotechnology, especially using recombinant DNA technology. But subsequent to that, probably right now more than 600 to 800 different products are in clinical trial. Probably about 60 to 70 pharmaceuticals, biologics are in the market, and many products are going to come. Please remember, altogether we have only 400 basic drugs are there, and right now about 10 to 15% is biologics, and this is going to significantly change to 60 to 80% biologics. That means recombinant DNA based products, whether it is antibodies, fabs, or sometime combined molecules, multiple targets, focusing on multiple targets. All these products are going to come. And from other few slides, I have mentioned about the new generation products, and there are novel innovations. Since I don't have time, I'm not going to mention, but you can see that absolutely super biologics molecules are going to come. And if I mentioned in the very first slide, we have developed a novel fab molecule using synthetic biology. So earlier biology slightly has changed. If you want to produce an antibody, you have to develop an antigen, inject into the animal, into mice or animals, and take the B cells and take the antibody, purify it, sequence it, identify the sequence, then you have to mutate. It's a very complex process. Now that has changed to a technology called fast display technology. That's what we are using. Now just see, modern biotechnology, it started from what? Probably microscopy is one of the earliest biotechnological tool people have used, because using microscope, you are able to detect, identify bacteria. The history of microscope. maybe 200, 250 years back. Even now, without microscope, you can't do anything. Then slowly, probably about 40, 50 years back, nucleic acid structure, function, replication is identified. What are the applications of this? You know about DNA, what it does, or central dogma, what exactly is central dogma, and transcribed to an RNA and translated to a protein and its application. Multiple applications using diagnosis, therapeutics, recombinant proteins. Almost everywhere it is now based on this biotechnology based opportunities with the DNA, RNA, or functional protein. Then gene expression. Now we know how to artificially express any kind of gene using a plasmid. That's what we are doing. Any kind of peptides, proteins, antibody, full antibody, part of antibody, combinations of antibody, or any of the types of protein can be produced using an expression system as well as a bacterial system, viral system, or fungus, or even plants, even in animals. So anywhere you are able to produce using the novel gene technology. Again, bioinformatics came. Nobody knew about bioinformatics. Now, using bioinformatics, using sequences, or using identifying the SNP, single nucleotide polymorphism, you are able to find out what is the disease, what is the disease profile, and the same technology can be used to identify how novel targets. Very interesting. That is called genotype phenotype analysis. All this require bioinformatics. Next generation sequencing, that after sequencing you need the use of bioinformaticians. Probably 25 years back, though India has got a lot of bioinformatics institute, nobody knew how to use it, but it is different. One of our company, MedGenome, employs about 250 bioinformatics scientists. All they have to do is in the computer. The same way bioinformatics can be used for docking studies. Either once the target is identified, the protein is identified, the structural aspects of the protein is known, you will be able to identify using the pharmacophore how this goes and binds to the protein. Recombinant DNA technology, which I mentioned. Now, where are the opportunities now? So, as I mentioned, the classical teaching opportunity, now it has changed multiple things, but these are the research opportunity. In agriculture, what all opportunities are there? Medicine and pharmaceuticals, food and feed industry, veterinary practice, environment, nanotechnology, and various other applications in life sciences. I can briefly go through this. So, if you really see crop improvement in various ways, the crops is productivity, disease resistance, or the quality, the quantity, many ways, everywhere. If you really see, the biotechnology has helped. So there is a few first generation traits in the crop improvement I have mentioned. For example, insect or pest resistance, herbicide tolerance, virus resistance, insect resistance, disease tolerance, and cytoplasmic male sterility. The other one is the second generation, like yield enhancement, nutritional quality enhancement, nitrogen use, water use efficacy, climate resilient genotypes, drought and salinity tolerance, heat and cold tolerance. Now, in the last 30 years or 40 years, plant tissue culture has come. Plant tissue culture, you will be able to culture the plant with an explant in the laboratory, and you will be able to control the quality. For example, you want dwarf or tall or heat resistance, or various opportunities have come. The same way, plant disease diagnostics as well as treatment can be possible. Treatment is using biopesticide. So what I wanted to tell here is that in agriculture, which probably we never knew about 40, 50 years back, except pesticide, chemical pesticides were used. Those things have changed now. Probably about another 20, 30 years, I think there is a very high chance nobody will be using chemical pesticides. Now just see the opportunities in medicine. Everyone knows about the chemistry or the pharmacology of drug discovery program. Now, the medicinal chemistry and synthetic chemistry and process chemistry is very, very important. Basically, developing the molecule. The first part of any drug discovery in NC is the synthesizing various types of thousands and thousands of molecules. Now, this has been slowly shifting into biotechnology arena, not fully, slowly started coming. Many of these molecules can be developed using biotechnology process. For example, simple alkylation process or nitration process, enzymes can be used. There are various advantages in using an enzyme. It is a green chemistry, the cost is less, high quality product simply by single step purification process. So developing NCEs is a big opportunity. Many pharmaceutical companies are already initiated the process. There are a lot of steps are used using enzymes. Now biologics, all the protein development, whether it is antibody, Fab molecule, or full antibodies, biosimilars, novel biologics, this is the core of the biotechnology. Probably the research is 60–70% is focused on this, mainly for the development of novel molecules. Novel drug delivery system, you all know how certain drugs can be tagged to antibody and can be sent. The same way drug delivery related to siRNA or short nucleotides to inhibit the RNA translation process. So these are some of the opportunities in medicine, and you all know about tissue engineering, how tissue can be engineered. If you have the cells, cultured cells, and if you have biological scaffolds, you can introduce that. So basically using the cell culture technique, you will be able to develop a total tissue. For example, artificial pancreas, that is one of the opportunity now, and many tissues similar way which can produce other enzymes or other functional proteins can be done. This is going to be very near future, and you know that, for example, in the heart valve is produced on a similar way. Gene therapy, how to inhibit an RNA, otherwise how a mutation, a lethal mutation can be removed. Now the science is great, it is a workable this thing. Right now people are working on that one, basically sending the nucleotides or or a primers or a short nucleotides into the area is not an easy thing, because the moment it enter into the circulatory system, systemic circulation, it will get hydrolyzed. So there is a tremendous opportunity also is there. Food industry, I don't think anyone has much heard about nutraceutical. Only in the last 20 years. That word itself is coined only 30 years back or something. This is a big industry in the US. Basically, nutraceutical do not require any FDA approval. But of course, in India, it is FSSAI approval is required. Nutrients or supplements that can be purified and given for certain minimal requirements. That is what nutraceutical. There are, for example, curcumin can be considered as a nutraceutical. Alpha-ketobospholic acid, that can be considered as a nutraceutical. So these are all mainly extracted from plants, but it can be extracted from anywhere. And you need to understand what exactly is personalized diet and nutrition. There is an area called nutrigenomics and nutritional sensors. Certain nutritional product can really change your phenotype. A very good example is fish oil, eicosapentaenoic acid. Eicosapentaenoic acid is known to significantly reduce the triglyceride level, and that's a drug molecule. So the same way, if you know that your genomics, based on the genomics, you will be able to develop a personalized nutrition. Food processing, in order to keep the fruits or other plant-based material intact, or extracting the juices and things like that. Lot of biotechnology application, majorly enzyme applications are there in order to preserve. And you all know about transgenic plants and animals. Transgenic plants like, Bacillus thuringiensis Bt cotton, which prevents the insects to attack on that one. Now, many such plants have come, many seeds have come, many fruits have come, but it is more or less, much less in India, but significantly you can see in the US market. The same way, transgenic animals for high production, milk producing cows, but mainly transgenic animals are used in the experimental purpose. And fermentation technology, various uses are there in fermentation technology. Veterinary practice, which was very poor in India, but that was not the case in the US. But things have changed. Animal vaccines, whether it is for chicken at the, the broiler chicken goes for only 40 to 45 days. And the seventh day, 3 different vaccine has to be given. Otherwise, all this chicken can die. The same way for other animals also, multiple vaccines are developed. Animal nutrition. One of the important thing is that how the nutrition can really be useful. There is a term used, FCR, feed conversion ratio, which is mainly used in the chicken industry, because chicken right now the cost may be 50, 60 or 70, something in that range. One chicken you can get for probably 200 to 50 rupees. Imagine 40 days, if you are going to feed the animal for 300 rupees worth food, they are going to eat probably in this, the chicken will grow up to 4 to 5 kg. And how much minimum food you can give? If you are going to give 100 kg, then it will never be possible. You can give probably about 8 kg food, which is costing about 20 rupees. Now, really, if you analyze, and there's an enormous opportunities there in the animal nutrition, using the normal food, how that can become highly digestible, how to improve the FCR. Breeding, there is again breeding, artificial breeding. There is an opportunity. Transgenic animal, which I mentioned, and this is diagnostics in veterinary, which is different from human diagnostics. So, and pets, companion animals, it's a huge industry in Western country, and it is coming in India too. Environment, and everyone talks about environment, how we can protect our environment, how our science can be used in the environment. For example, biofuel, biodiesel, bioremediation, that how environment can be protected using our novel technology, especially growing trees and fungus, etc. Wastewater treatment, huge number of liters of water is going as waste containing unwanted thing, either whether it is fecal matters, unwanted bacterial or household products, even drugs, chemicals, how this can be treated. It's a very big industry, especially using biotechnology. For example, microorganism. There is various ways are there. Microorganism identify using carbon nitrogen as sole source, identify the right bacteria to kill this such chemicals. That's another opportunity. Biopesticides and green chemistry. Briefly, I told green chemistry is that use of proteins or enzymes to convert one pharmacophore into another pharmacophore. Nanotechnology is one of our core area. We have developed several products using that, as I mentioned, using nanotechnology. Probably it can come under biotechnology or material science or chemistry, but everyone considers nanotechnology as a part of the biotechnology. Whether it is for drug delivery or imaging process or diagnostics, or in wastewater treatment, or nanorobotics, which is futuristic thing. If you see, all other 4 things we are working on, and many of our products are already in the market too. So nanotechnology is another interesting area, companies where you can also work on. Life sciences, mainly research applications, whether it is for forensics, gene editing, or development of biosensors, or synthetic biology, which briefly I mentioned, and bioinformatics. Synthetic biology is basically use the synthetic understanding. You can develop anything. Just understand one thing. Using synthetic biology, you will be able to make the gene. You can make the cell. That means, for example, including cell membrane, which is fatty acids or lipids, or other proteins, everything can be produced and you can develop a theoretically a live cell. That is basically synthetic biology. Now, if some of you are interested to have some startup idea, I'm not mentioning about the biotechnology, biopharmaceuticals, which you know whether it's connected to drug discovery, diagnostics, and other things. There are many other opportunities are there. So diagnostics, then agri clinics are there. For example, if you want to test the soil, because not everything will grow anywhere, so you might be able to set up small such facilities everywhere. You can develop what are the nutrients which is deficient in a particular place. You can test the seed. Is this the right seed? Is the genomes is right, or this is resistant to pests? Those type of things can be tested. Then post surveillance, then diagnostics and control services, etc. Green chemistry is
Sir, you are
another.
not audible now.
Oh, since how long you are not audible? I'm not audible. Hello. Oh. 1 minute. My microphone is on. Can you hear now? Hello.
Yes, sir. Now it's audible, sir. No problem.
Okay. Please tell me in case. But could you hear all the things what I was telling? From since how long you did not hear? Hello. Hello.
No, I was hearing.
Since how long you did not hear? Can you hear now?
Yes, sir.
Okay. I removed that. There is a microphone connected. So, yeah. Yeah. Please let me know in case you can't hear.
Sure, sir.
Yeah.
Now it's audible.
Okay. Since how long you did not hear? Okay, I was telling about some simple startup ideas other than biotechnology. Yeah, hello.
Hello, sir. You can continue. There's a small technical issue in the department. Okay. You can do the rest of the audience. And
Sorry, I could not hear. It was not really audible.
okay, there is a small technical issue in the department who is hosting the program. But the rest of the audience they can listen. So you can continue.
Okay. Yeah.
Thank
So
you.
in case there is a problem, let me know. I'll immediately.
Sure,
but,
sir.
I mean, yeah. Okay. I was telling
Sure,
about
sir.
some simple startup ideas. What are the things people can do? Biodegradable plastic production is one area. Probiotics is another area. Making platelet-rich plasma injections for various opportunities. Aloe vera production plant. These are some simple ideas which I thought I will just put. Not mentioning about the biotechnology, core biotechnology, which is recombinant protein or drug discovery or diagnostics. I'll also mention how much more time I have. I can continue, no?
Yes, sir. You can continue. No problem.
Yeah, I will
10
briefly.
or 15 minutes. You can do.
Yeah, I will briefly also discuss about the entrepreneurship in life sciences. So
That's wonderful.
Yeah.
That's great. That's great.
Yeah, many entrepreneurs has come up in the last probably couple of decades, and you can see that's if you really see the statistics, you'll see that, not much has happened from India. I know all of you know about Amazon or Facebook or Apple or Microsoft or Tesla. So the owners of that, who has developed that one, first time entrepreneurs. And if you really see, there is some unique characteristics in that one. Before that, let me explain to you who is an entrepreneur. There is various kind of definitions, explanations are there. Some of the interesting ones, someone who envisions, creates and executes an idea that they are absolutely crazy about. Very, very, very important. They should really think that this is something working. Because Tesla, the electric car producer, he also thinks that people can be sent to the moon or sent to other planets or even to the so that is one of the interesting thing he is planning. And you know about what is Microsoft, this is all because of Microsoft. Or Apple, I'm using only Apple computer and the thing. Facebook, I think is a part of everyone's life now. The same way Amazon, I think all over the world. So it's very interesting. That's all came how many years? 10, 20, 30 years. And these are some of the richest people. What they have done, some novel idea they got, they converted that into some products and really useful. And below that, first person, he was my boss, Dilip Sanghvi. For a while, he was the chair, the richest Indian. He is still one of the richest Indian, fourth or fifth richest Indian. Dilip Sanghvi, owner of Sun Pharmaceuticals. He's only a BCom person. And he started with 10,000 rupees, but amazingly brilliant and, a visionary. He has a vision and he follows up things. Can you imagine? And when I was interviewed, I was amazed the way which he interviewed. A BCom second class person. And you know who is the second person. He's from Kerala. I hope you know. Dr. Verghese Kurian. Dr. Verghese Kurian is the father of milk industry, not only in Gujarat, probably all over India. And many, many countries follows his idea and vision. And third one, of course, everyone knows. Ambani, the father Ambani, what he did. It's like, he was working in a petrol pump from where he has come. His son has become the richest, one of the richest in the world. Now, what is tying up? I have just showed that, a person is tied up. Entrepreneurship in India, you really see there is actually each part really can be explained one by one. Since time is not there, I'm not giving. But why many people did not start entrepreneur or did not become an entrepreneur? I myself started as an entrepreneur only last 5, 6, 7 years. This is not the time to start companies or anything. You should start when you are 20, 25, 30s. It is worthwhile. And if somebody has any plan, don't delay. You will go through some hardship, but it is worth doing. Now, these are some of the interesting, unfavorable things in India, unfavorable marriage market, because people think that person do not have money. That's why he started his industry. Lack of respect. For example, if he's a doctor, fine. If he's a doctor who is an entrepreneurship, no. There's no respect. Educational loans. Another major problem is that, many of the students have got a lot of loans. So somewhere or other, first 10 years, they want to work. They have to work because they have to pay the fees. They have to take care of their home. Then stay-at-home spouse. That's another big characteristics in India. Wife has to stay at home. Husband only has to work. That will not work. If both of them are there, one of them can really start the entrepreneurship program. Academic block, because I don't think when I was doing my PhD, nobody told about how to start a company, what are the opportunities, nobody, nothing. And mindset of the society which mentioned that, entrepreneurs probably is like a business person or like a shop owner. Fear of failure, yes, that is, I understand, because 90% of startups fail. Mainly scientific startup. Even restaurants start, it fails, 90%. Please do not worry. The reason is that once you have entered that, nobody will consider that as a negative. That failure, you have learned a lot. With that experience, you can further improve. But Please remember, everyone's background, if you see, it's a big failures can be seen. But once you have entered put your foot into that, you will not come up, because you will continue as an entrepreneur. There is no startup advice over there. By the way, in Trivandrum, Inter-University Biomedical Center, then, the Kerala governors, Prime Minister, Chief Minister has there is a program is there. I'm an advisor there. Then in RGCB, there is a program in, this Sri Chitra Institute, all these places I am involved. But right now there is multiple places. It's kind of started. But we can't see too much of enthusiasm like what you can see in the US. There is every year, there is 1 million new entrepreneurship is programmed. Only 300 million working population is about 150. Huge number is doing. In India, it is 1,200. Imagine. So these are some of the tying up factors. Now, if you are going for a entrepreneurship program, please consider these 3 P's of startups is very important. Let me also tell you, there is nothing like to unravel the complete human potential, nothing like starting a startup, because it's not doing a small job. End to end, from setting up a company, naming the company, to make it commercially viable, it is not an easy thing. There will be about 100 different tasks out there. I have not seen anything so complex as setting up an industry, making it successful. You will be working 24 hours, but if you are having that passion, you can do that. And if your profession, like for example, a doctor starting in a hospital, or a scientist starting a biotechnology unit, and with all these things, you need to have a purpose. If you are thinking, oh, I need a job for that, no. You need to have purpose. Your purpose could be like, I want to become one of the largest industry. Like Byju's he has started. Nobody knew about this online educational program. He is the richest Keralite, I think. Probably 4th richest Indian. Imagine a simple idea, how that is translated. What is required? You need to have passion for that, whatever you are doing, and your profession should be the passion. And the purpose should be there. You need to have the purpose not for making money, but support your country or your state or your people, whatever it is. Now, another 5 essential elements of success. This is a very interesting survey was done about 100 failed company, 100 successful company, including Microsoft and all. And people think that, if you have money, everything is there, you will be able to do. It is not that. One of the most important thing is the right team. A team who do not fight with each other. That's the most important thing. And the second most important thing is timing, the right timing. For example, Infosys started about 25-30 years back. That was the time mainly the IT industry initiated. So timing is very important. The team, if you see Infosys, there are 5 dedicated team members who are there, non-fighting. Then other things are there. You need to have a good idea, business model. All the other things are there. What I found is that money is the easiest to get. If you have the right team, good people to work with, and the team building is very, very important. There are bonus points that you need to have a good networking skills should be there, and the team should have a very good skills of what you want to do, absolutely good attitude and adaptable situation, because you can't you may not get huge amount of money and all when you are starting a startup, but you can expect that after a few years. These are the cycle, or is like how to start a startup from ideas. Then there is a planning, there is a teamwork, then identify investment. Then what are the trends in the market? What are the resources you need to get? Then you need to have the right strategy, the product development, and you can commercialize. This is the cycle. But it's not as simple as you think, but it is worthwhile doing. Now, there are 3 things before you start working on any startup. Be super passionate about your startup idea. That's very, very important. If you don't believe, don't do. If you 100% believe, whatever it is, you will be able to do that one. And such very interesting idea you can get, whether it is biotechnology, biopharmaceuticals, pharma area, life sciences, agri-foods, any of the things, interesting ideas. And please do that. There's a lot of possibilities are there. And don't think that you will be able to work from 9 to 5 and 4 days or 5 days. No, it will not work sitting at home. And if you are married, your spouse has to be 100% supportive. Otherwise, things will not work. Be ready to accept failures. That's the most important thing. If you think that you are going to be failure, make sure that you have a support system when you fail. Like your wife or husband, somebody should be working. Or you need to have the sufficient money to take care of your family. But I can 100% tell you one thing. Once you have entered an entrepreneurship program, whatever the job, whatever is the salary, wherever you get, you will never go back. You'll continue with the entrepreneurship. But I can definitely tell, eventually you will be successful. You go back and see any of these industries. They would have failed enormous number of times. This is the startup roadmap. Now, if you really see, each of these, what I mentioned, point has something or the other to do. If you see the top part, finalize the idea, incorporate the company, then secure intellectual property, secure local licenses, workplace, initiate operation on full scale. It has only come up to this point. Then regulatory approvals, commercialization process, with all these things, identifying the team. So quite a lot you have to do. For example, finalize the idea, you need to study the market, rough business plan, team should be ready, name of the company. People think that it is very easy to get a name. I will tell you, it is not easy at all. You can see some so funny names because such names should not be there anywhere in the world. If I cannot start like, Microsoft. Immediately, I will get the notice from a lawyer. I can't change the spelling and put. For example, I thought Microsoft Life Sciences. No, not possible. So please remember, even the naming is very important. Incorporating the company. There's a lot of process, various types are there, but it is best to do as a private limited company. Then you need to have, if you want to get sufficient money, you need to have the IPR should be important. Local licenses. You might be amazing. GST, PAN, MSME registration, Startup India registration. There is local manufacturing license. Like that, many things are there. So these are some of the points you will have to look into, and how to pitch to an investor. That means if you are going to an investor, how to tell him your story. Please remember, don't use like what I am talking 1.5 hours. Nobody will, then immediately people will stop at that point. You need to give everything in probably 10 minutes. And there is something called elevator pitch. The very first talk will be 30 seconds and maybe 3 sentences should be there. What you are planning to do. The investor immediately understands the importance of that. Then after that, you tell the problem. Then what are the technology solutions available? What is your business model? Have you got an IPR, or when you are planning to file? Then definitely a regulatory slide, especially life sciences product, everything has got a regulatory slide system there. Then competition as substitute. Please make sure if too much of competition is there, nobody is going to fund. That's why if you want to start a restaurant, nobody is going to fund you because enough and more is there. But if you are starting something risky, it can be risky, but returns can be 100 times, 1,000 times. There will be enough and more investors will be there. You need to study market opportunity. Leadership team is very important. Then how to, if somebody is investing, how to exit the investors. There's something called IPO. That means after some time, that investor should get, for example, if he have put $1 million, he is expecting 3 million to 5 million in 3 years' time. How he can get it? What is the method? Then you will have to see what are the potential risk mitigations. Every business, every startup has got a risk is there, and make sure that you give a very good summary of that. Then some of the startup success. I'm not going to explain what is it.
because of the limited time. Please remember the Airbnb, very interesting, a multi-billion-dollar company now. Started about 15, 20 years. Genome is our own success story, our own company. Very first time started, next generation sequencing. Multiple investors came, valued at 400 plus million, and is expected to make it to $1 billion company in few years' time and go for an IPO. I will also mention about some of the product what we developed, anti-VEGF, age-related macular degeneration, an antibody or Fab antibody. Fab means fraction antigen binding. AMD means age-related macular degeneration. So this is the molecule, an antibody, but what we have done is that part of the antibody. And we used fast display technology and used recombinant DNA technology, expression system, fermentation process, purification, analytical development, regulatory. It took 5 years, but very successfully we have done. One of the best molecule probably might come out because it is far superior than the existing molecule, which is called Lucentis. That's the important thing. Now, another one we are working is on, this is is all about wet AMD, how it happens. There is a disease called AMD, age-related macular degeneration. 80% has got dry AMD, which is vision loss, very difficult to see. There is as such no medication, but 15% become wet AMD, and the problem is that you won't be able to see. Total loss of vision. Imagine at 50s and 60s, losing your vision is a horrendous problem. And this is another molecule which we are developing for diabetic retinopathy. The similar type of molecule, anti-VEGF molecule, but the mechanism of wet AMD and mechanism of diabetic retinopathy is very different. But similar type of therapeutic approach. Right now, only 2 molecules are there, and we are developing a much better molecule. In the next 6 to 9 months, it is expected to reach the clinical evaluation. Another company we started right now, 2 months back, with a company called Prodigy in the US. So this is Theragen molecular innovation, mainly developing IgY molecule, which I mentioned. Now, instead of fast display technology, fermentation process, what you need to do is that give the injection to chicken and layer chicken. They will lay egg. You can take the egg yolk, and from this you can extract the immunoglobulin. And right now we have already developed such technology mainly for alcohol-induced hepatitis. People who drink extremely high, they can get hepatitis. Hepatitis is mainly because of a bacteria which is outgrown other microbiome called Enterococcus faecalis. This Enterococcus faecalis also has a molecule like endotoxin called cytolysine. This can paracellularly permeate into the systemic circulation and enter into the liver damage, hepatitis. We are developing a first-in-class molecule against this cytolysine and the bacteria. Mac genome, probably I might have told earlier, this is one of the technology which I have developed probably 20-25 years back when I was in UK, but translated in the last few years back. Many PhDs did with me, whether in UK or in India, and all the technology is very different from others' nanotechnology because we are using naked particle. Everyone till date uses coated particle because no one knows how to use the naked particle and bind other molecules to that. That's what we have done. It's a simple process visualization. For example, DNA extraction. So we are going to all over India to universities to use this one. We started selling, but of course now it has all came down to zero. But we'll have to pick up again because of this corona problem. But we have that opportunity we used and started making other products for corona. So these are the products of what we have developed, and many products are in the pipeline, including various automation process. SIP-wise is we are developing certain products for them using SIP-wise. What we are doing is that functional drinks, various functional drinks, We have developed a turmeric shot. The advantage is that this turmeric has got high bioavailability. Then immune booster, turmeric, large quantity we are making. Morning aid, hangover drink. So many people who drinks heavily has got hangover. Against that, because we developed such a things, there are not many competition, not many such products are there. Amor, a sexual wellness drink, mainly that, those who have got less libido. So this is something activating the certain systems to the sexual desires. So that's another 2 products for males and females. Then another product what we are developing is a beverage, initially starting with a beverage against COVID antigen. Spike protein antibody we have developed, and this will be an OTC product. So the product is ready. Now we are waiting for the approval. This product will go all over the world. Subsequent to that, we are going for oral nasal spray, the same type of product, but different composition. Finally, mouthwash, because these 2 products, as an OTC, it is difficult to introduce because in India, it is controlled by DCGI, Drug Control General of India. On the other hand, beverages are by FSSAI. So this is the details which I have given, how we are going to do the OTC products to contain the spread of COVID virus. Now, skill set developments in biotechnology, biotech as an industry. It's a young industry, especially in India. Very long and costly time cycle. That is something which you need to understand. You can't develop a product overnight. The risks are very high, especially if you are involved in recombinant proteins, getting the approval. A very simple example, India has not approved a single novel NC or biologic till date. Most of the molecules are coming from US, but we have approved a lot of biosimilars. Very volatile, dealing with unknowns, human health and disease. Of course, any drug discovery program is complex. And you require highly specialized labor pool. This is something we are facing tremendous problems. So that's why we have a training program. First, we train between 3 months to 6 months, but we are facing a problem also. After 6 months or 1 year training, they do not want anymore to work in a startup. They go to companies like Syngene or Biocon. So this process is going on. So this is some areas what we are facing some problem. Now, what is the current position of biotechnology in India? Maturing industry. Slowly it is growing, but not at the pace what you can see in Europe and US. High student inflow. Maturing operational talent. Fundamental knowledge of disease growing. More clinical trial know-how. Biotech established itself as a valid partner and innovator. Area of chronic need. Then spirit of innovation and entrepreneurship, agility embodies industry. Stock performance increasingly more stable. So I'm just telling what is the current status, current position of biotechnology is a slow starter, but slowly picking up. The major problem, single problem, if anybody asks, it's not the money. Team development is not the people, but skill set people. It's very difficult to get the right people. And even to train, it is not that easy. So this is one of the problems we are facing. How to break into biotech, leverage skill set, take tiny steps, get in with your skill set and then move laterally. Find a company that has biotech practice area, network. So if you want to get a job in this area, I would suggest the first thing what you have to do is your skill set, certain skill set. And you should be able to answer the questions which is normally asked by the industry. I can tell you one thing, till date, 99% people do not know how to make a 0.1 molar phosphate buffer. Now, because that's one of the requirements, the answer is that it is there in the internet. Now, this type of understanding is required. Like, if how an assay is done, how an enzyme, how to do a KM and Vmax, many people are not able to do. So these are some of the problems that we are facing. Then identify a career path you want. Do your own research because there are a lot of opportunities to get into a job. Internet job sites are there. professional and trade organization website, biotech industry news, career opportunities, biotechnology. So these are biotechnology, Toby Freeman. So this is a networking site. Okay. So, but what I suggest is that, the most important thing to have a real network with the people, and 80% of the positions are filled by this networking. For example, if you know very well your professor, they can suggest such people. And I will tell you, most of the industry in this area is by, you will never see an advertisement in a paper because you can never get through headhunters. It doesn't work. Networking is the best thing. Otherwise, directly write to the HR, briefly, one paragraph, why you want such a job. But also remember one thing, whatever you have put it in your CV, you should know. If you don't know, don't put it. If you have put certain things technique, go and study very well. Because the interviewer is not going to ask something which you don't know. They are going to ask what you know. As I mentioned, networking is very important. You can get it from your own organization, meetings, informal interviews, local community and friends. So, and volunteer, professional organization. One possibility is that sometimes some of the companies, you just go and volunteer. In US, it is a big practice. Volunteer. That means you will not get anything. They will get all the other, whatever is required. You will get that maybe 6 months and all whatever is required, computer facility, internet, everything we will give. But now these 6 months, they study. And if you are good, you will be immediately absorbed even before that. We do that very clearly. That somebody really will see for 3 months and the supervisor comes and tells, this person is very good, we'll normally take. And attributes for success at a company. How you can be successful. But let me tell you, your attitude should be extremely good. That means others should like you. And you should be a team player. Very, very important. Now, your team lead is telling something, you go opposite, go on arguing. It doesn't work. You have to follow one single step. Whoever it is, your reporting person is very, very important. Just follow him. Then only things will work. Otherwise, what will happen, if it is in 20 different places, no product execution will take place. So this is something we are focusing totally on. Very good attitude. They should know how to be a team worker. They should follow their boss. Then will be fine. Ability to become interested in wide variety of different scientific areas. Learning is a continuous lifelong experience. Please remember, anybody in research, you can't stop reading. You can stop attending seminars, continuous reading, be part of discussion. And the most important thing to be a very successful research, your analytical ability. For example, if the data is there, or when you are doing an experiment, something goes wrong, where it is gone wrong, and how that can be sorted out. Communication skill. I'm not telling about very high quality English, but you should be communicative, not keeping quiet. And presentation skills is very important. That not some of these slides I've given probably is not the right way. You need to have minimal write-up, and just by seeing one single slide with picture, the person should be able to understand. This is a different kind of talk. That's why I'm giving. So don't follow this type of some of the earliest, early I have given is the right type of slides. A lot of pictures should be there. By seeing, they should be able to understand. Contribute your particular talent and expertise towards the common goal. Success means that your project grows so that hundreds of people work on it. But with all said and done, I can tell you one thing. Try to find a place where a startup company is started by some really highly educated, a good scientist who knows the technology. You will be fine. And too much of rules, regulations, like 9 o'clock means 9 o'clock coming. If you are 10 minutes late, you will lose half a salary. Don't go to such company. These are called Lala companies, that you won't even know what you are doing. Unless the first thing is that, if the company is so confidential about what you are doing, don't go, because you need to learn. So these are some of the things you have to learn. And please make sure that if you are ready to work for 2 years, tell that you will be there for 2 years. That's all. That's fine. Nobody will bother after that. But don't tell that I will be here forever, 3 years and 6 months you are leaving because you are getting a better job. That will not work. So there are various career opportunities are there. Like, some very interesting, something different, immunoassay, sample analysis from human clinical trials. This is some of the things which you can even start. Formulation development, you can support the formulation development for other companies, mainly for pharmaceutics people, and even for, if you know about nutritional product can be formulated. There are not many people. Pharmacology, assessing drugs in animal models. Animal model development is very important thing. Protein sciences, cell line generation to overexpress recombinant protein. Protein characterization, biochemical, biophysical characterization of a protein. We have only few places in India to do that one. For example, the configuration and conformation of a protein, because if somebody wants to do, there is tremendous opportunity. Assay developments. This is many companies are failing this one because they don't know how to develop a good assay, and the people also do not know. Assays means functional protein. Is the receptor assay or enzyme assay or transport assay. This is very important for whether it is biologics or small molecules. Protein manufacturing process development. That means recombinant protein, the expression process, how to ferment, then how to break out the cell, and how to purify, how to analyze, how to find the configuration conformation. Program coordination and management is another thing, because you can also go, if you are not interested to do lab work, you can always become a manager to manage other people, but you need a lot of other requirements are there for this one. Then, manage core facilities, methodology oriented, DNA, insight to facts, mass spec, biocore, etc. Then clinical evaluation. Then regulatory, understand FDA guidelines, license for company to FDA, European Union. Regulatory understanding is a big thing. The same way patent writing, scientific writing, and quality control, managing, quality assurance, and business development, sales and marketing development. So these are various area career opportunities there. Then how to build your resume for an industry in R&D position, which I have briefly mentioned. Focus on accomplishments and achievements, focus on results, provide facts. As I mentioned, please make sure that don't put so many things, what you know. And many times I have seen that they will put something, when you ask questions, they don't even know the word. They never ever do that. If you are putting that you know how to do recombinant DNA technology, transformation process, or how to assay an enzyme, learn everything about that. Throw out the standard academic CV, describe yourself, use key buzzwords for HR. Make both a single page and full version of your resume. Bring both versions to all meetings. Make sure that in one page or half a page, everybody will get some idea. Give the full version with the prompted or to the hiring manager. Make sure you build a cover letter and make it specific to the job. References are very important, because many companies take only after reference, especially your teachers and your superior. Tell the person that you are applying, and you can tell your employee that please call such and such people. I was under that person, or my PhD grade or whatever. It is very important if that person gives a good reference, great. But please remember, no professors, no guide, no managers will a bad reference. If the person is very good, they definitely will tell that person is very good, all the positive qualities. If the person is telling, okay, like that, then you assume that the person is not very good. So how to apply? The basic method of applying for an industry position, job fair, which I have already mentioned. There are various ways you can apply to the industry. But as I mentioned before going through that, mainly networking is very important. Make sure that you have an extremely good network. Thank you very much, and you can